Hi, good day to all. In this video, we'll be seeing about the AVERY experiment. So, it is an evidential full stop for the search of genetic material. So, it is actually the continuation of Frederick Griffith's work. So, it was mainly done to explore the biochemical nature of the transforming principle. So, it is actually a search for the genetic material or the biomolecular nature or a biochemical characterization of transforming principle. So, now we'll have to look into that. The continuation of Griffith's work was done by the trio team in 1943. Oswald Avery, Macklin McCarthy, and Colin MacLeod. They continued the work of Frederick Griffith. So it was done mainly to determine the biochemical nature of Griffith's transforming principle in an in vitro system actually. They used the phenotype of the same Streptococcus pneumoniae strain, so which was used by Frederick Griffith, expressed on the blood agar in order to figure out the transforming principle. So we shall look into the experimentation. In this experiment, the trio team they have used the cell extracts of heat-killed S-type cells, that is smooth colony forming, virulent type Streptococcus pneumonial cells. First of all, it was made sure that the cell extracts was free of lipids and carbohydrates. Then it was divided into three sets because it was treated with three different kinds of enzymes. So we'll see one by one. So the first one, the cell extract, it was treated with protease enzyme. The protease, as we know, they are protein digesting enzymes. So when it was treated with the cell extract, so automatically it would have degraded all the proteins within. Now, the extract which is free of protein was made to transform the R-type cells, that is rough colony forming, non-virulent Streptococcus pneumonial cells. So as a result of transformation, we could find S-type cells on the plate. So what does this result indicate? This indicates that protein is not the biochemical nature of the transforming principle, right? Why? Because if protein is supposed to be the biochemical nature, transformation could not have occurred. It would have inhibited transformation. So other than protein, there is some biomolecule which is responsible for transformation. That is why here, without any regret, the transformation has occurred, right? So the second set of cell extract, the extract was treated with RNase enzyme. The RNase, it is supposed to be the RNA degrading enzyme. So it would have degraded all the RNA within. So now this RNA free cell extract, it was made to transform the R-type cells. Again, rough colony forming, non-virulent Streptococcus pneumonial cells. So as a result, transformation has occurred. As you can see, it has resulted in the formation or in the colonization of S-type cells. So the transformation of R-type cells has occurred in this case also. So which means the RNA is not the biochemical nature of the transforming principle. So that is why it has not affected the transformation. So here we can find the S-type cells in the plate. And the third thing, the third set of cell extract, it was treated with DNase, which is supposed to be the DNA degrading enzyme. Now, it would have degraded all the DNA, as we know, because the DNase is an enzyme, it is a DNA digesting enzyme, so it would have degraded all the DNA within the extract. So now this extract was made to transform the R-type cells. Again, the same thing, that is the rough colony forming, non-virulent Streptococcus pneumonial cell. As you can see in the plate, only the R-type cells was found. So the key, it is here. DNA, it is the biochemical nature of the transforming principle. So that is why the transformation has not occurred, right? Because in the cell extract, it is free of all the DNA. So as there is no DNA, there is no transforming principle here. So that is why it has affected the transformation. So through this experimentation, it was concluded that DNA is supposed to be the biochemical nature of the transforming principle. So now continuation to conclusion. So through experimentation, it has been so evident that the treatment of cell extracts with protease and RNase, it did not affect the transformation, but digestion with DNase has inhibited the transformation. So, which means that the DNA is supposed to be the biochemical nature of the transforming principle. So, it was concluded that the DNA was the genetic material or the hereditary material which is responsible for transformation. So, that's what I said, like it is a continuation of Griffith's experiment to a conclusion or a evidential full stop for the search of genetic material, because only through this experiment it has been proved that the DNA is the biochemical nature of the transforming principle. Thanks for watching. Happy learning. Have a good day. Thank you.